Welcome. Welcome back to Facts in Biology, a YouTube channel that is devoted for the preparation of NEET and many other medical entrance examination. Friends, through this channel, I am continuously attempting to provide you with the in-depth information about NEET syllabus, CBSE syllabus as well as the state board syllabus. Myself, Professor Anand Joshi from Malegaon, MSG College. And I hope if you find these videos helpful for your preparation, please do subscribe, like and share and strike the bell button so that very easily you can get notified whenever I am uploading a new video. Friends, today we are going to discuss a very important concept in the angiosperm. A post-fertilization process of development of endosperm is the topic that we are going to discuss today. Friends, we know as a result of double fertilization, triple fusion, there is a formation of two important things. The first one is diploid zygote that consequently develops into the embryo, and the second one, triploid primary endosperm nucleus. That is the second thing that is produced as a result of double fertilization and triple fusion. Now this triploid primary endosperm nucleus, it undergo repeated mitotic proliferative divisions, and these mitotic divisions, it gives rise to a nuclear multicellular nutritive tissue that is said to be the endosperm. Now this endosperm is the post-fertilization tissue which is produced as a result of post-fertilization processes. As a result of that post-fertilization process, diploid embryo and triploid pain, they are seen to develop side by side simultaneously. Now actually the other cells of the embryo sac, they undergo deorganization, they are degenerated sooner or later as they are no longer useful for any further development. Now the formation of triploid endosperm nucleus, it triggers cell division, mitotic cell division, which leads to the formation of what we call it as an endosperm. Now there are the different patterns of development of endosperms, and generally there are 3 to 4 types of endosperm development are recognized, and these three types, these are main: the nuclear, the cellular and the halobial type of endosperm development. Each one we are going to see in detail, and additionally towards the end we will have one more additional method for the development of endosperm. Now the first method, it is said to be the nuclear type of endosperm. Now this nuclear type of endosperm, it is the most common type of development of endosperm in angiosperm. Almost 161 families of angiosperm, they show nuclear type of endosperm development. Here the primary endosperm nucleus, it undergo repeated mitotic proliferative divisions and without formation of wall. So as a result of that, large number of free nuclei are seen floating in the endosperm, and these, we can say, nuclei, very soon they move towards the periphery and there is a big vacuole which is created in the center. If required, later on, these nuclei, they undergo the second, we can say, part of mitotic division, that is said to be the cytoplasmic division, or we can say cytokinesis. And after cytokinesis, there may be formation of cell wall, cellular plate, so as to form the multicellular endosperm. But in several cases, cell wall formation or cytokinesis, it is not observed at all, or it remains incomplete at the time of maturity of the fruit. Most commonly it is found in the plants like wheat, sunflower as well as in coconut. A very interesting thing about the coconut that we can say is that coconut, as we know, it contains the white flesh as well as the water, coconut water we call it as an. Actually both of them are representing the endosperm. The whitish part that is lying towards the periphery, it is actually nothing but the cytoplasmic, or we can say it is a complete cellular endosperm. And in the center, the liquid part, actually that liquid is nothing but the nucleoplasm of the different nuclei, large number of nuclei which are present within the coconut. The second type of endosperm that we are going to discuss, it is nothing but the cellular type of endosperm. In some plant, division of primary endosperm nucleus is immediately followed by cell wall formation, cytoplasmic division, or formation of cellular plate. So that endosperm is cellular right from the first stage. Here you can see continuous division is there and there is a formation of cell wall between the resulting nucleus. As a result of that, it is, we can say, continuously cells can be observed in the endosperm. Now it is mostly found in the plants, around 72 plants of dicots, and most common example are balsam, petunia and adaxa, and many other plants are there where cellular endosperm can be observed. The third type is there, it is called as halobial type. Now why it is called as halobial? Because it is generally observed, for the first time it was observed in the plant belonging to family Halobiaceae and the order Halobiales and we can say superfamily Monocots. In this case, the first division of the primary endosperm nucleus is followed by a transverse wall formation, and that will result into formation of two unequal cells. Here you can see there is a formation of unequal cells are there, and these unequal cells will continue to develop into the endosperm. The smaller cell which is produced towards the chalaza, it is the chalazal cell, and the other cell which is present towards the micropylar end, that is said to be the micropylar cell. Now the nuclei in each cell, it divide by free nuclear division, proliferative division, and then there is a formation of cell wall between nuclei right from the micropylar end. Now in this mechanism we can obtain or we can observe a dual kind of development, or it is said to be the intermediate method of development of endosperm, which is intermediate in between cellular and nuclear type, and most commonly it is found in the plant like Asphodelus. Now this Asphodelus, it is nothing but the plant that is very commonly observed in the European countries. The third, or we can say fourth one, which is additional type, it is called as mosaic endosperm. Now this mosaic endosperm, it is a we can say endosperm which is containing tissues of two different types or two different, we can say, pattern, and as a result of that, these two different type of tissues, they are arranged in a mosaic design, and that's why it is called as mosaic endosperm. In plants like corn, endosperm contain patches of two different type of tissue. Here you can see the darker one and the lighter one. Now these are the patches of tissues which are produced during the development of endosperm, and as a result of that you can observe a mosaic design in the endosperm. Friends, if you find this video helpful for your NEET preparation, you please subscribe to, like or share my channel Anand Joshi Biology. Thank you very much.